Trepka from Harvard, position-specific attachment of nanoscale samples.
Okay. Hi, everyone. My name is Kai. I work in the group of Dr. Tao, who talked earlier about the silicon electron transfer properties. So our group as a whole is concerned with imaging mostly the smaller class of things. We have lots of tools to look at big things like cells and bacteria. We can use optical microscopes. We can look at some slightly smaller things. We can try to look at viruses and proteins with things like SEM, but unfortunately, electron beams often destroy biological samples. So this is where scanning probe microscopy, which is the point of this whole conference, comes in. And there are even some images down to single atoms. I believe there's a group that imaged a benzene ring and several similar aromatic molecules. So the technology in particular that our group is interested in is a version of magnetic force microscopy called magnetic resonance force microscopy. But sort of in its dumbed down form, MFM looks and works a lot like this. And Professor Wong talked about this earlier, so I'm not going to go over the exact details of how a measurement is performed. Just know that you need a magnetic tip to interact and measure the magnetic response from the sample. There's an input sine wave and an output sine wave. And then you can extrapolate that into measuring the magnetic properties of a sample. So like, for example, the sort of dummy MFM graphic on the last slide would produce a lined and ridged image like this, which is from Park Systems. So there are two common setups if you want to do magnetic force microscopy. You can either place or any sort of magnetometry. So you can place the sample under the magnetic cantilever or magnetic probe and sort of run the probe over the sample and measure the magnetic response. The other thing you can do is you can place a defined magnetic field below the rod attached to your sample, and you can scan the sample over this defined field and also get a magnetic image of the sample. So these two setups have sort of different challenges in terms of how we want to approach them. So if we're thinking about the second one, the sample-on-tip magnetometry, one problem you might think about when imaging biological samples is how do we get them onto the tip? So our first impression would be, oh, they're bacteria grow in colonies. We'll just dump the tip in the colonies. Some of them will stick on by van der Waals interactions or by crawling onto it, and then we can kill them and image them. So this turns out not to work. Christian Deegan attempted this measurement, and it took a lot of effort to sort of get a small enough number of samples on the tip to do any well-defined measurement. It turns out that if you're trying to do a bulk attachment of samples via van der Waals interactions, you get a ton of samples at once, which makes it very difficult to image a single sample, which is often what you're interested in terms of performing any quantitative imaging. The second problem you run into, which I'm sure many of you are familiar with, is probe tip convolution. This is an AFM image I took of basically a bunch of nanoparticles. I had crashed the tip into the surface earlier. It was a bunch of triangles, so I got a bunch of triangles. This is a common problem not just for sort of silly mistakes like this, but also for AFM and scanning probe microscopy in general, which is that if you have a very large tip, the shape of the tip becomes confounded with the surface of the sample, and it's unclear which is which, and it's very difficult to get a precise spatially resolved image. So I've just told you about two very different problems, maybe that you have differing levels of familiarity with. The question then remains, what do these have in common? Why would I bring them together? And the answer is the motivation of this talk, which I call a position-specific linkage, or a position-specific attachment of a nanoscale sample. So the sort of model problem I'll be discussing here is attaching a tiny tip to a slightly larger tip so that we have better spatially resolved magnetometry. The sort of broader view of this kind of process is that you could also hope to singly, covalently attach a biological sample to a tip if you were interested in magnetic force microscopy. So then we wonder, well, what's the point of this? So if we look at a general MFM setup, we have to have some sort of probe with a magnetic tip scanning over a sample. And if we zoom in on this probe, in general, if you buy a commercial probe, it'll have a relatively large tip. Someone earlier mentioned theirs was 100 nanometers. What might be nice, if we do an image with this, it might look a little blurry. It's unclear exactly what this is, if there are well-defined dots, if they're connected or not. But if we instead make a nano heterostructure and then attach a small tip to our larger nanoparticle, we can hope for a better sort of spatially resolved image, which might give something a little better like this. So then the question comes, how do we do this? How do we go about creating a position-specific linkage? So the route that we've started on in our group for developing this kind of probe and this kind of method is one that combines physical and chemical methods. But we start with something relatively simple, which is a carbon-coated magnetic nanoparticle. The reason we choose a graphene coating, as several people earlier have elucidated, is that graphene is very good at preventing things from getting in, which means that nanoparticles, which are often subject to oxidation, especially at small scales, which ruins their magnetic properties, can be effectively protected by a graphene coat. Fortunately, so after we have this graphene nanoparticle, we then run into a different problem, which is that graphene is, for the most part, non-reactive, which makes it so useful. So we perform a cyclization reaction to attach a functional group to make graphene both soluble and usable so that we can modify it in the future. After we have this carboxylic acid group, we can perform an amide alkyne click reaction, which is well studied as the basis of protein formation, to attach another partner to it. And then essentially we go on attaching chemical groups until we get to the gold, which has a high physical affinity for sulfur chemicals. So step one of this was getting a graphitic nanoparticle. Fortunately, there's some published work on how to go about doing this. So I won't go through the exact details, but basically you get a bunch of iron atoms in a silica matrix. You perform CVD to coat them, and then you remove the silica matrix with an HF etch. And at the end of all of it, you get these sort of ugly nanoparticles because the parameters aren't fully tuned yet. And then fortunately, you can buy slightly prettier commercial nanoparticles from a company online. The problem with the commercial ones is that they are slightly large. They're a diameter of 20 to 30 nanometers. So if we want nanometer level spatial resolution, we may need to optimize the other fabrication route in order to achieve that. The second step is covalent functionalization. Lots of people have worried about dispersion of graphene and covalent functionalization of graphene, how you go about attaching different groups to it. Fewer people have worried about covalent functionalization of these graphitic nanoparticles. The most common route is that people will just shove them in a sonicator with some very long fatty acid chains and then hope they get stuck. Often they do get stuck. But we've decided that in terms of thinking about the strength of the bond, we would prefer a covalent interaction. So there's this literature-reported reaction where you can incubate graphene with this perfluoroazide. So we decided the chemistry is probably the same with this graphitic nanoparticle. It has the same reactive azide group and the same sort of alkyne bonds to react with. So maybe we should try this and see if we can get it to stick. The answer is yes. It seems that we can covalently functionalize the graphitic nanoparticle using the literature reaction that was published in the context of graphene. It demonstrates increased solubility in DCB, which we expect now that there are fluorine atoms on the product. Most importantly, we have an IR spectra showing the development of a CF2 stretch in the product, but not the reactant, indicative of the attachment of this perfluoroazide functional group. Unfortunately, this IR data is very recent, so we have not done any further modification of this particular chemical, which means that this portion of the talk, this set of chains, is largely over. The other thing we sort of need to complete this experiment is the gold at the other end of it. To create a dummy gold substrate, which we can think of as a gold nanowire attached to a silicon nanorod, or a gold nanoparticle attached to a silicon nanorod, you can also just put a gold nanoparticle on a surface and say, well, that's a pretty good model of it. If it sticks just to the gold nanoparticle and not the surface, that's an indication of specificity. So we make this dummy surface just by spin coating commercial gold on silicon. And it's pretty monodispersed. So for the next step of the project and for checking attachment, we can compare the location of gold particles with the location of these larger particles and make sure that they stick in a one-to-one fashion. So this is all a pretty annoying chemical strategy where we have to transform, or we have to create graphene, we have to transform it into one partner and then another click partner and then another click partner and then perform these high specificity reactions. So one thing one might consider is, isn't there some sort of alternative, easier physical strategy? And actually there is. So instead of doing this whole complicated process, you could think of, let's just get our magnetic nanoparticle and grow the probe off of it. So something like this, maybe. This was actually touched on earlier by Professor Zhao. So Zhao's work focuses largely on thinking about this in the context of colloids, which are large micron-sized particles. But in theory, glancing angle deposition should work just as well with a small particle as a large particle. You get a small particle. It'll go on a substrate. You tilt the substrate. And then atoms will impact it and preferentially grow into rods off of the small particles. Our hypothesis is then we can detach these nanowires, which are now silicon nanowires with a magnetic nanoparticle on the tip. And this can function just as well as sort of the spatial probe that we were thinking about earlier. Then your next logical question is, well, does this actually work? So we have some decent data that indicates that this might be working. This is just a substrate with a bunch of nanoparticles on it. Doesn't look very meaningful. We shoved it in the evaporator, and we evaporated a bunch of atoms thermally, silicon oxide, to try to grow the silicon lever arm off of our magnetic particles. This is the result. So it indicates that you can actually get shadowing and growth. The important question is, are there actually nanoparticles on the tips? Our preliminary answer is, it seems so. There are clusters of bright, distinct spots in the SEM, which often will indicate elemental contrast. The better answer is we can't be absolutely sure without TEM characterization of these products. But the preliminary information indicates that, yes, you can use nanoparticles in a similar way that Professor Zhao uses his colloidal nanospheres to grow things off of them and develop heterostructures that are nanostructured for the purpose that you want. So in summary, MFM is important for analyzing very small objects. Several other members of this conference have used it as such. We seek to optimize it by developing better attachment methods and have pursued both physical and chemical methods that, while preliminary data exists, require further modification. And then I want to thank my group, two of which the members are here, and also Park Systems for this conference and for their excellent technology, such as AFM, that enables us to intermediately characterize our products.
Open for questions.
Yeah.
So what's the lifetime of that functionalized probe tip that you can grow this way? How long would it last for you? Is it mechanically strong? How long does it?
Right. So which method? By the Glad deposition or Glad deposition? We haven't seen them decay or break down yet. So we don't know the lifetime because they haven't stopped working. We also haven't used them as probes yet. We've just physically created them, but it's not like they spontaneously decay. They have very long lifetimes.
Can they be used to image within a liquid-solid interface?
Within?
Rather than just ambient?
I'm not sure about that. Yeah.
In your second method of growing it, what kind of radius were you able to achieve sharpest?
So the dummy particles we used as the tips here were the ones I grew, so they were between 50 and 100 nanometer graphitic iron particles. But in theory, this practice will, or this shadowing should occur regardless of diameter, which is an experiment that we're currently running.
One approach you showed was coupling the graphene-coated nanoparticle to a gold nanoparticle and then to a tip?
Yes. So the rationale, or go on, what's the question?
So why not just use a gold-coated tip and then use thiochemistry to couple to this graphene particle? Why have the gold nanoparticle?
No, that's the same thing. The gold tip is equivalent to a gold nanoparticle. So like often when you're using VLS growth of silicon nanowires, you'll seed it with gold. So it's just a model system for that sort of process.
So this isn't the assembly?
Yeah, so you're exactly right. That's exactly what we're doing. We have a gold-tipped probe, and we're trying to attach a nanoparticle to that gold tip.
I have a short question for you. Would you happen to know the cohesivity of the ion particles to start with?
We measured it, and if I remember, it was around 50 Oersteds.
So it's very soft.
Yeah.
Thanks. Any other question? Let's move to that. Thank our speaker.